Greetings, grade 12s. Okay, we're going to be doing some revision questions based on meiosis in this video. So please make sure you've actually gone through the videos relating to meiosis or you have revised this. And also, same that I did with the DNA, please make sure that you've actually done these questions. Because remember, meiosis is quite a complicated section. So you need to make sure you know what you're talking about. You need to actually do the questions to gain the most benefit of doing this revision. What you may notice is pretty much almost all the questions, bar one, are actually taken from previous IEB examinations, the sort of the long ago ones from there. So it really is a good idea of the kind of questions you're going to be getting. So let's dive straight into it. So I've got question one. Consider the following image of a cell in one particular phase of meiosis. So here's my picture of my cell. So here's my questions. So question one. Identify the phase of meiosis occurring in the above picture. So by the way, if you haven't done the questions already, pause, do the questions as we go along, and make sure you do them. So identify the phase of meiosis occurring in the above picture. Well, clearly what I can see is that there are homologous pairs that are sitting next to each other. There's like some touching of things that are happening here. It's like one cell. The nucleus is kind of still there. So clearly this is prophase 1. So you have to say the 1. You can't just say prophase. And again, you can either use an I or you could have said prophase 1 with the actual number. Really, that's actually fine. So that's what that's what this picture is from there. Then it says label structure A. So let's go into our picture and we see, okay, A is the bracket going over these 2 chromosomes that are here. The structure, I'm looking at both of them. Those are homologous chromosomes. Okay, nice and easy so far. Then I go label process B. So again, I'm in prophase. There was a couple of things we needed to know happen in prophase. But if I'm looking at B particularly, it's a process, not a structure that I'm labeling. What is the process that's occurring there? And that is crossing over. Ooh, too many S's. Crossing over. The exchange, again, so it says label the process. If you come and say a sentence like, it is the exchange of genes where one touches another, no. It said label. So the label, the process is called crossing over. Same with labeling structure A. We are not sitting and saying, these 2 chromosomes are the same length and carry the same gene. No, it is a label. Label it as homologous chromosomes. You have to make sure that you read the instruction and understand what the instruction says. Okay. Then it says, describe the importance of process B. So we're talking crossing over. We have labeled this. Why is process B very important? Well, the exchange of genes, so don't tell me what it does. So I'm not asking you, is it an exchange of genes? No. Why is that important? Because it creates genetic variety as our first point. And then what is our follow-on from that? Because you see it's for 2 marks. So genetic variety is the 1 mark. Why is that important? So you pretty much have to then have some link towards evolutionary success or being able to resist environmental change. Could be that we were, something along those lines. That's what we're looking at for an evolutionary success, being able to resist those things from there. So it was 2 marks, 1 thing, genetic variety, then our link from there. Okay, nice, easy first question. Then, question 2. Which phase of meiosis is being shown in the picture below? Justify your answer. Okay, so we're both labeling what the phase is, and we're justifying. So let's look at what we've got in this picture over here. So the way that you want to try and identify is look at the chromosome. So the first way that you can tell is either how many cells are being created, but in a picture like this, you have to be careful because you can't just assume, oh, it's going to be 2 cells, therefore it must be phase 1. No, we can sometimes show you a phase 2-1 only from 1 cell, and that is what's happening here. Why? Because if you look at these actual chromosomes, what are they like? What do they look like? Well, first of all, we can tell that's an anaphase. First of all, we know that it's an anaphase that's happening because the things are splitting. They haven't reached the end. They're not lined up on the middle. There's definitely a split that's happened. But is it phase 1 or is it phase 2? So this is where it becomes the justification part. So what is actually separating here? Again, look at that table of differences between meiosis 1 and meiosis 2 of the anaphases. In this picture, the sister chromatids are being separated, not homologous pairs. Therefore, it has to be anaphase 2. So that's the 1 mark for which phase. Now to justify your answer, why is it 2? Because sister chromatids are being separated, and that's really your reason. And you could even say not homologous pairs. Show that it's your understanding. Rather give more, even though it's only 2. Even if you just said sister chromatids are being separated, well, what does that mean? It means it's not homologous pairs, meaning anaphase 1. There's a separation, but it's the chromatids that are being separated. Okay, I hope you got that one. Again, look at what the chromosomes are drawn as. The drawing of the chromosomes is very important in meiosis. Okay, now another interesting one. The following diagram shows a pair of homologous chromosomes in prophase 1 before crossing over. The letters represent genes, and the numbers represent potential points of crossing over. Potential points. The chromatids are numbered 1 to 4. So we've got letters that are genes. Do we notice the difference? These are small letters. These are big letters. Sort of linking back to that picture that I showed you in the video when I talked about this process, crossing over. The numbers are where it can potentially link. And this is the chromatid number. So, question 1. Name the process which caused the corresponding genes of each pair of each chromatids to be exactly the same. Okay, so break that question down. Name the process. Okay, so it's a process, not a structure that I'm doing, which caused the corresponding genes of each pair of chromatids. There's your key word of chromatids, the corresponding genes. So if I'm looking chromatids, I'm looking between 1 and 2 and 3 and 4, not across the homologous pair. So why is it that these letters are then the same as these letters? Why is it that these letters are the same as these letters? What process allowed for that? Well, that is, let me just get my colors going, DNA replication. Because remember, DNA replication is part of the meiosis process. We learned this although we did it by protein synthesis, we learn it as part of meiosis. So the reason why these are exactly the same is because we duplicated them. DNA replication. Okay. Assume crossing over occurred at point 7 of chromatids 2 and 3 only. Write down the new genetic sequence for each chromatid after crossing over has occurred. Nice little difficult question here. Okay, so the question says, crossing over occurred at point 7 of chromatids 2 and 3. So where's chromatids 2 and 3? 2 and 3. So this chromatid and this chromatid did crossing over at point 7. So this here is where the chiasmata was happening. And this is where they are going to switch their genes. So everything below is going to be switched. So, what will the new genetic sequence be? Well, the easiest ones is to do 1 and 4. Because, why? They don't change, because they didn't touch. It was only chromatids 2 and 3 where the crossing over happened. So 1 stays exactly the same as the original. So it was A, B, C. So it was A, B, C, D, E, F, G, H, I, J, K. Where does it go up to? Up to K. And then on 4, it's exactly the same. It's just capital letters. A, B, C, D, E, F, G, H, I, J, K. Right. So those are the easy ones. Then, now we get to the one where we had the crossing over. So the difference is the crossing over happened here. So literally, the last two are going to switch. So it's going to be, let's get my color, A, B, C, D, E, F, G, H, I. Then it will be, no, I. Sorry, I just realized that shouldn't be a capital I. That should be a little I. And then it will be J, K, because they switched. And then it will be, with my red, A, B, C, D, E, F, G, H, I. And then little j, little k. So that's where showing where the crossing over happened. That's the difference in the genes that's being created there. Okay, so that was the crossing over one. That's a slightly more difficult, slightly more applied question, but you do need to start getting used to potentially having those questions being asked of you. Okay, I'm going to pause the video. I'm going to stop the video here, and I'm going to do a part 2 for this. So make sure you go back over those, make sure you understand, and then we'll see you in part 2.